And now we welcome online Ruidong Xiang. I'm sorry for the pronunciation, I'm not sure. Yes. Good evening for you. Good morning for here. Ruidong is a scientist in Melbourne, at Melbourne University and DPI, in the very well-known group who invented genomic selection. And I will speak about regulations mainly. Ruidong, the floor is yours.
Thank you very much. Good morning. I hope you can hear me. And it's an honor to be invited by Didi and the Wolverine Consortium. So my name is Ruidong, and so I just recently started as a new start at the Agriculture Victoria Research. Today, I want to share with you about our research on to identifying regulatory variants to enhance mapping and prediction of cattle traits. So it would be a bit of mixture of science and industry utilization. So I think the previous speaker already discussed that most of the traits in mammals probably are complex. So that means they're very polygenic. So this is a graph taken from human GWAS catalog. And basically says that for about 9% of GWAS hits fall into genic region, and that means 91% of the GWAS hits are in regulatory regions, and that suggests that cognitive trait loci should at least overlap or impact gene regulation. So you'd think something like a SNPs affecting gene expression or splicing. But in fact, there has been quite a debate on whether this sort of EQTO or SNPs that affect gene expression, how much genetic or phenotypic variants contribute to the complex trait heritability. So in human studies, it's known that this is a puzzle, that about 11% of human trait heritability due to expression QTO regulatory variants, just a small fraction. In cattle, we previously evaluated there was also around that number, and there's a limited overlap between cattle QTO and regulatory variants. But our latest study published last year show something quite different. So this is a collaboration with the GTEx consortium, where we really looked at large-scale transcriptome or EQT mapping data, so a sample size approaching 5,000 across 16 tissues. And we mapped trans and cis expression and splicing QTOs. And then in a different set of 120,000 cattle, we partitioned our SNPs into 7 classes. So we got trans cis and trans E and splicing QTO, plus another set of random SNPs. And we use these classifications as Bayesian prior fitted a model called Bayes RC that allow us to partition the trait heritability into these categories. And as an average across the 37 traits that we evaluated, we found that the cis, E, and splicing QTO explain about 45% of the trait heritability. So this is an average across 37, so varies for individual traits, but certainly the standard error is small. About 24% are due to trans regulatory SNPs. And this is, we suspect this is largely due to the good sample size in EQTO mapping data set. And shows the case that the regulatory variants are quite important for phenotypes. So this is a little breakdown of that amount of variance explained on the y-axis across different tissues. They are similar, there are differences, but the amount of total variance explained by EQT or splicing QT are similar. But at the very end, there's a multi-tissue category, which is the SNPs that affect gene expression or splicing in many tissues or across 16 tissues. And what's happening here is that when we only look at the expression QT alone, for single tissue, there is about less than 10% variance explained across different cases. But when we look at the multi-tissue, even just the EQT alone, that amount of variance explained of phenotype or heritability is bumping up to 50%. And if we put E and SQT together, then at a single tissue level, we explain more heritability. And then at the multi-tissue level, that gives us the largest fraction at around 70% of the trait heritability due to E and SQTO. So apart from that, showing that a lot of heritability can be explained by regulatory variance, it also shows that if you include more regulatory variance, then you tag more heritability of the traits. So we took another set of traits to do another, kind of validation. So this is called lipidomic traits, but essentially there are 56 lipids measured from cow milk using mass spectrum facility at our group. And we apply the same method to partition heritability again. So this time, the y-axis is the difference between heritability explained versus expected if they are just random SNPs. So you would expect that if they are important, they should always explain more than zero, which is the case. So, and in this case, we see the transvariants. So the SNPs that are distant to the genes explain more variants of the lipidomic traits. And because each explain a bit more across these categories, then they all add up. In total, they're about around 70% of heritability in these lipidomic traits are also due to the regulatory variants. And here is an example. Obviously, we prioritize the list of these important or potentially causal variants in the paper, but this is an interesting one. So this graph shows the result of this region in base R. So it's a clean result showing that this top variant is significant for birth size. The PP is not super high, but in this case, anything that's above 0.25 is considered important. So there's a clean result here. This variant affects birth size in cows, and it's located within this gene called IRAG1. And this gene is, according to Ensemble, annotated as missense, and it's also at the site that is conserved across 100 vertebrates. And so this variant in the other trait that is, sorry, I forgot to explain this, is from chromosome 15. That affects the concentration of this lipid in the milk. So this is another graph, but analyzed by base R. And we also looked at its expression QTO. So this variant is a cis expression QTO in the blood. So it actually affects the expression level of the neighboring gene called CTR9. So it's quite interesting that this SNP, which is missense for IRAG1, it is actually affecting the expression level of its neighboring gene. So that's something quite new we haven't seen before, and we have quite a few cases in our study. So that's in potentially interesting causal variant. So we find many important SNPs that we certainly want to use them into our genomic evaluation. So we have this pipeline that, or life cycle of designing commercially available SNP chip using discovered SNP chip SNPs. So obviously we do lots of research, we'll find and validate some interesting causal variant, and then we'll go through this life cycle. So we'll start to design a chip and do the manufacturing, and then we're obviously going to do imputation in the reference and in historical new animals, and then we do genomic evaluation in the industry, and if they're all passed, we'll get on to get the commercial chips. So this is our current SNP chip has gone through a cycle for at least 3 times now, as I remember. And so the latest version, we give it a breakdown of the composition of the SNP chip or customized SNP chip. Have very small amount of mitochondrial SNP, and again, very small amount of SNPs tagged genes. We tried very hard to put about 2.7% of indels on a SNP chip. They were very difficult to deal with, but we tried. So it was good. We obviously had a fraction of the SNPs that we had from the old chip, so we put them on. And then the majority of the SNP markers are from this sort of research and discovery validation process. So we've certainly had the industry in Australia, Datagene, adopt this customized SNP chip. So this chip now is extensively used, and it's been used to genotype and evaluate about 20,000 cows, dairy cows. It has been implemented for Australian Holstein cattle and also some native Australian red. We're working on a process for Jersey, so it should come. And we have evaluated this system or customized chip for many times. So this is one of the latest studies. This is by Iona, McLeod, and Irena Vandenberg. So basically, the y-axis shows the prediction accuracy, and it's a comparison between the standard chip and the customized chip. Now, the blue bar is the accuracy if your evaluation is in a population that's distant from the reference, and the green bar is that the validation population is close to the reference population. Now, you know that if you do a genetic evaluation in a population that's distant to the reference, then you will have a drop. So obviously, you saw the blue bar is smaller, shorter than the green bar. It means there's a drop when your evaluation population go distant. But when you use the standard chip, the drop is larger than using the customized chip. So, you know, the drop was about 12% on the standard chip, but the drop was 8.8% on customized chip. So in a different angle to support that there is a bit of robustness in this customized chip. Obviously, it still is drop. You would like to see there's no drop, but that will be working in progress. Maybe we'll include the epigenomics chip markers in the future. So future steps. Now, you probably already feel that this sort of putting markers on a SNP chip is quite tedious. It could get tedious. So I know you spend a lot of time, get these prioritized SNPs, then you go through the SNP chip manufacturing process. Some couldn't pass this process, and you have to go back. And some, when you made some and they couldn't pass the genotyping QC, et cetera, and you always do new research and you work with new traits. You know, you have methane, you have heat tolerance, you have some environmentally important traits. They are coming, so you do new GWAS, you find new variants, and you have to go through this scan again. So that would be not ideal. So we are moving towards to use low pass sequencing to replace SNP chip. Obviously, we're talking about the industry applications. Costs is the thing. It's not quite at the price as SNP chip yet, but obviously it should come down over time. So we are preparing for that. So that's one of our moving directions to use the. So then, when you have a set of prioritized SNP chip, you don't design a SNP. If you have prioritized SNPs, you don't design a chip. You gotta just go back to your low pass sequencing and just impute by weighting some of the prioritized SNPs. The other moving direction is to look at the long read sequencing. These are so Iona Tuan and Amanda are the people who to contact if you're interested in joining them. But the reason to look at those is to have structural variants or complex indels or other variants rather than SNPs to be included in a routine evaluation process. Obviously, this is not even close to industry application, but like the previous speaker, that we're also looking for something that just not just SNP genotypes, but it could be something in addition to SNP on the genome that can help us track more heritability, so that we can increase the prediction accuracy and speed up the genomic selection process. And that's all, actually. So I'd like to thank people give us data to analyze data gene in Australia and CATO GTEx Consortium, and the collaborators, obviously our computational team, Australian and Research Council, and many other collaborators. And this is the past and present computational biology group. And thanks for their help. And that's all. Happy to take questions.
Thank you very much, Ruidong. Any question in the room or online? Not online. In the room?
There is one online I can see. For low-pass sequencing approach, will you use only the variants that were initially on the SNP genotype in SNP chip, then imputation or all variants? That's a good question. I'd probably say, so to do the latter, all variants, then we have to have low-pass sequencing to do the training, which means that we have to use that for 200,000 cows. That's probably not realistic to start with. So maybe the former, which is we will use the SNP chip genotype and impute to sequence, and then the next step we'll use the low pass to do the prioritized SNPs. I hope that answers your question.
Christa?
Sarkun, FLI. Thank you, Ruidong, for this excellent talk. I have a question regarding the large efforts you take to really find or pinpoint SNPs which have a regulatory causal potential. And on the other hand, then you go for imputation. Have you checked whether, or is there a danger that with this imputation process you lose information on those low frequency but highly effective regulatory SNPs? Have you checked this? Because on the one hand, you're really trying to dig very deep and pinpoint single variants, but then you go back to imputation. Have you checked how much you can lose?
Yeah, so there are a couple of things here. One is that the reason that we're doing this is because so if you normally do imputation, you start from just ordinary SNPs. So we want to start imputation from the informative SNPs. So if we know some SNPs are already important for dairy traits, then we would like to impute from there. So we will lose some, but if we have prior knowledge that they are important and we impute using them, then it's better. But second point is, yes, we do lose rare variants for sure. It's a matter of cost effectiveness versus the accuracy. Now, whether or not rare variants tag or enrich more trait variants than common variants, that itself is a debate. In the humans, they say it's not. Not a lot more enriched than in the rare variants than common variants. In the animals, I believe we haven't really tested that, but we did something still based on imputation. We didn't see that the rare variants attack more variants than common variants, but that study itself is confounded by the fact that the rare variants are also imputed. So they are not important, potentially because they were not well imputed. So I don't have the answer for you, but I would like to be proved, or would happy to be proved wrong that the rare variants are still very important or miss them, and we would like to capture them as well. It's just that very pricey to genotype.
Yes.
How do we manage the trade-off between, sorry, Chris, or it's CRV. How do we manage the trade-off between selecting SNPs that improve imputation accuracy versus selecting SNPs that have the potential to be causative? Because as we were discussing a second ago, you kind of lose something by targeting SNPs that are in regulatory regions or regions that we suspect have a large effect size, but at the same time could possibly gain more by choosing SNPs for imputation, knowing that we're going to lose something in imputation anyway.
That's a really good question. Well, I guess we could. So when we do the variant prioritization, when we're trying to select for causal SNPs, we did cut out the SNPs that are imputed poorly. So in that case, we've filtered out the possibility that, well, not filter out. We kind of merge that information for imputation quality into this SNP prioritization project. But we haven't done a quantitative test to see that, to what extent if we prioritize imputation accuracy could get us to a better imputation. So I don't have the answer for you, but it's an interesting area to look at, I believe.
And so I have the feeling that we reach rapidly a plateau when you already have, let's say, 20,000 SNPs, good SNPs for imputation. You gain very little, adding 5 or 10,000 more. So, and this extra room can be available for candidate genes. I have another question. Is your chip targeting a number of traits? And is it suited for additional traits you may have in the future? Do you need to find the new regulatory variants to add them, or is your chip already okay for the future traits?
Not quite okay. So we do need to find new SNPs that associate with new traits. So that's why we are tired of making the customized SNP chip again. That's why we have this intention to switch to the low pass sequencing, so that we don't have to make another chip, but we might have to do new imputation from the low pass again and again. So yeah.
Okay. And in the genomic evaluation, do you have any special procedure to account for these markers by weighting them differentially, or do you just use GBLUP?
In the industry scale, it's mostly done in GBLUP because that's large data size. In research type or research-ish industry setting, we could use base R or base RC. So we would have fit this different annotation as prior to increase the prediction accuracy. But industry scale, I believe it's mostly GBLUP.
Okay, thank you.
Yep.
Any other question?
There's a question
Online, okay.
So did you compare low-pass sequencing versus GBS, genotyping by sequencing approach? Not on my top of my head. Maybe my colleagues have done it that I am not aware of. I'll let you know if I get to know that. Thanks.
Okay.
I can answer that if you like, Ruidong. It's Amanda.
Yeah, go ahead, Amanda.
Yeah. So we have done a test of low pass sequencing. I'm quite happy with that. We're currently testing GBS, targeted GBS assay that targets this, I guess, this panel, this custom panel that Ruidong's been talking about. We think we can get that down to a similar cost, but we actually think that we will land on something that's a compromise between the two. So a low pass sequencing with the targeted small number of targets, and in particular, these ones that don't impute well, spiked in GBS as well. So that's where we're headed. We hope we'll be there in about 6 months.
Thank you, Amanda.
Okay, I would like to thank the speaker warmly, and please join me.